Dear student, today we are going to discuss about medicinal chemistry 3 antibiotic chapter. Now, this particular chapter is included in the semester 6 of the medicinal chemistry third subject. So the first topic today we are going to start, that is antibiotics. This is lecture number 1, in which we are going to cover historical background, nomenclature, stereochemistry of that particular compound, structure activity relationship, chemical degradation and classification of the particular category, that is antibiotic. Myself, Dr. Ketaki Dhane, here going to explain everything in detail about antibiotics. If you have not subscribed yet to this channel, please subscribe so you will get the notifications when I am going to upload the every new video. Thank you. So we are moving towards the antibiotic chapter. Basically, what are these antibiotics? They are any substance which is produced by the living organism. So antibiotic हा एक असा कंपाउंड आहे, that means which will produced by the living organism, and that compound has a capacity to inhibit the growth and the survival of the other species of microorganism. That means substance produced by one species of microorganism have the capacity to kill or to inhibit the growth of other species of microorganism. That particular compound we are named as antibiotic. And very important terminology or very important concept under this category is those antibiotic will show their action in very low concentration. That means antibiotics in low concentration can inhibit the growth of other species of the microorganism. Now the historical background behind this discovery of the antibiotic. First, in 1877, Louis Pasteur discovered the inhibition of some of these species of microbes by other microbes during when he is doing his research work on the anthrax species. Followed by in 1908, a very long time, Gelmo synthesized the first sulfonamide drug, that is sulfanilamide. Now this sulfonamide is the category of the compound specially used for the antibacterial derivative, and this is synthesized from the similar compound which is one of the cellular component of the microorganism, that is PABA, para amino benzoic acid. From this PABA we are going to synthesize the sulfonilamide or sulfonamide. Followed by in 1910, Paul Ehrlich discovered the selective strain of the microbes, that is antibiotics, or we can say the magic bullets. He nominated the drugs as a chemotherapy or the chemical knives. Eisenberg studied in 1930 bactericidal properties of the azodes with the sulfonamide. Now the bactericidal property is any antibiotic or any chemical compound which, if he is going to show the bactericidal or bacteriostatic property, bactericidal means totally killing of the microorganism. That means this compound will kill the microorganism, and bacteriostatic means a chemical compound will stop the bacterial growth. Cidal means killing of that microorganism. That means Eisenberg studied the properties of azodes which will create the along with sulfonamide. So this is a combination therapy. Azodes plus sulfonamide can kill the microorganism, that is a bactericidal property. And if any antibiotic, any compound, if it is going to stop the bacterial growth, त्याला काय म्हणलं जात आहे, bacteriostatic. That means antibacterial compound is divided into two categories, bactericidal and bacteriostatic. Followed by in 1928, the discovery or accidental discovery of the penicillin. It is happening and it is discovered by the Alexander Fleming. In 1932, Ehrlich discovered the antimicrobial activity of the sulfonamide. So in 1932, in 1908, Gelmo synthesized the sulfonamide, but in 1932, the Ehrlich discovered the antimicrobial activity of the sulfonamide. In 1943, the drug companies actually started the synthesis of penicillin and production of the penicillin. In 1948, a second category of the antibiotic came in picture, that is synthesized by the Oxford University, and the name of that second category of antibiotic is cephalosporin. Followed by in 1952, erythromycin came in picture, which is actually an aminoglycoside type of antibiotic, which is synthesized by Streptomyces erythraeus. Followed by in 1956, vancomycin introduced the penicillin resistant species. Now, no doubt the penicillin is used to treat various types of infections, but these compound has its own limitations. Penicillin is degraded immediately in our acidic as well as basic pH of our body, digestive system. So in order to overcome this acidic and basic pH degradation penicillin, vancomycin come in picture, which is introduced and which is resistant towards those microorganisms which are resistant towards the penicillin. Next, in 1962, quinolone antibiotic were discovered. Followed by in 1980, fluoroquinolones were discovered and become clinically useful. The example of the chloroquinolone compound is ciprofloxacin, which is used as an antibacterial agent. And in 2000, a new category, that is macrolide antibiotic, introduced into the clinical practice. Now we are moving towards the first category of compound, which is discovered in 1928, having a magical therapy, can be used to treat various types of infections. They are of bacterial origin. What is the history of penicillin now? Now, penicillin is discovered by the Alexander Fleming in 1928, who noticed that one of his experimental culture, now he is doing his study on the Streptococcus species. Now during his study, what happened? One of the mold or one of the culture is contaminated with the mold or the fungi. New category of the mold or fungi is contaminated the Streptococcus species his strain. Now due to this contamination, the contaminated mold lies or broken down the species which are there in the that broth. So what happened due to this contaminations? This mold belonged to the family of penicillin. This penicillium mold or the penicillium fungus contaminated that broth and done the lysis of that broth or done the lysis of those bacteria. Now the fungi is from which origin? The name of that fungi is from Penicillium notatum origin. So he has given the chemical derivative name as a penicillin, and this penicillin he further on extracted and derived from the species of the mold, that is Penicillium notatum. Alexander got a Nobel Prize for this particular type of discovery, and this is the chemical extract. He further continued his study in order to extract the penicillin from Penicillium notatum to stop the growth of other microbes. Further, there is discovery of various types of penicillium takes place. The penicillin V, penicillin F, penicillin G, and followed. Further, in Florey and Chain introduced the penicillin in the therapy of chemotherapy, and Waxman in 1942 given the elaborative definition of the antibiotic. What is actually antibiotic? Already we have discussed. Compound produced by living organism have the capacity to kill the other species of the microorganism. They are of chemical substance or the drug which is produced by the living organism or marine sources and can inhibit the growth of other microorganism. Highlighted word, which is very important one, in how concentration or on in how much concentration? In very small concentration. That means antibiotic in very small concentration can kill the microorganisms in the higher concentration. They are not effective against, very important one, this category of compound. No doubt they are antibacterial, no doubt they are antimicrobial, they have the capacity to kill the microbes, but they are not useful against viral infections. They also includes they are derived from some sort of synthetic origins, some sort of semi-synthetic origins, and so on. So depending upon a variety of uses, their chemical nature, their mechanism of action, what type of chemical structure they are going to have, antibiotics are basically classified into 4 categories based upon the spectrum of activity, based upon the source of origin, based upon the mechanism of action, and based upon the chemical structure. Now first classification based upon the spectrum of activity. असे antibiotics की जे particularly useful or particularly going to stop the growth of ओनली सिलेक्टिव्ह स्पेसीज ऑफ मायक्रोऑर्गॅनिझम, म्हणजे एका ठराविकच मायक्रोऑर्गॅनिझमची ग्रोथ किंवा त्यांना ते मारू शकताय अशा अँटीबायोटिक्सना आपण कॅटेगराईज केलं स्पेक्ट्रम ऑफ ॲक्टिव्हिटीच्या अंडर. जर ते एकाच इफ दे आर गोइंग टू kill or to stop the growth of microorganism towards only selective species, they are narrow spectrum of activity. And if we are going to use for both gram positive and gram negative, they are known as broad spectrum of. So this is depending upon the spectrum of activity. Next is based upon the source of origin, whether they are derived with ते कशापासून उत्पन्न केले, फ्रॉम विच कंपाउंड दे आर डिराइव्ड, whether they are obtained from the natural origin, whether they are obtained from the semi-synthetic origin, or whether they are totally synthesized in the lab. Third classification is based upon the mechanism of action. Through what type of mechanism of action they show their antibacterial properties, whether they are inhibiting the cell wall synthesis, whether they are inhibiting the protein synthesis, whether they are interfering with the nucleic acid functions, or whether they are interfering with the functioning of the cytoplasmic membrane. So depending upon these variety of mechanism they are classified. And fourth is what type of chemical structure they have, they are classified. Depending upon the chemical structure they are classified into beta-lactam antibiotic, aminoglycoside type of antibiotics, tetracycline type of antibiotics, macrolide antibiotics, and miscellaneous derivatives. So we have discussed that is based upon the spectrum of activity, if they are classified as narrow spectrum antibiotic and broad spectrum antibiotic. If they have a selectivity or high degree of selectivity towards only a single species of microorganism, they can use to kill the stop the growth of only single species of microorganism. They are narrow spectrum antibiotic. And broad spectrum, if they inhibit the growth of gram positive as well as gram negative microorganisms, so we are categorizing them under the broad spectrum of antibiotics.